Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be going through the article, A Physical Wiring Diagram for the Human Immune System, by Shilts et al. From the Wellcome Sanger Institute in Cambridge, UK. This paper was published on August 3rd of 2022 and was honestly in a review for about a year, June 30th, 2021 to June 28th, 2022. So we'll just get into it. The human immune system is composed of a distributed network of cells circulating through the body, which must dynamically form physical associations and communicate using interactions between their cell surface proteomes. Despite their therapeutic potential, our map of these surface interactions remains incomplete. Here, using a high-throughput surface receptor screening method, we systematically mapped the direct protein interactions across a recombinant library that encompasses most of the surface proteins that are detectable on human leukocytes. We independently validated and determined the biophysical parameters of each novel interaction, resulting in a high confidence and quantitative view of the receptor wiring that connects human immune cells. By integrating our interactome with expression data, we identified trends in the dynamics of immune interactions and constructed a reductionist mathematical model that predicts cellular connectivity from basic principles. We also developed an interactive multi-tissue single-cell atlas that infers immune interactions throughout the body, revealing potential functional contexts for new interactions and hubs in multicellular networks. Finally, we combined targeted protein stimulation of human leukocytes with multiplex high-content microscopy to link our receptor interactions to functional roles in terms of both modulating immune responses and maintaining normal patterns of intercellular associations. Together, our work provides a systematic perspective on the intercellular wirings of the human immune system that extends from systems-level principles of immune cell connectivity down to mechanistic characterization of individual receptors, which could offer opportunities for therapeutic intervention. So moving on to the introduction of this paper, the human immune system must maintain the same coordination and cohesion as the body's other homeostatic organ systems, despite being composed of highly migratory and circulating cell types that are distributed throughout the body. Diverse arrays of cell surface proteins organize immune cells into interconnected cellular communities, linking cells through physical interactions that act both for signaling communication and for structural adhesion. The immune system has been described from one perspective as carefully coordinated networks of cell types, where by extension it is these physical linkages that hold the network together. Consequently, immune receptors regulate virtually all stages of cellular activation and are appreciated as critical mediators of a variety of homeostatic and pathological processes, which range from tumor surveillance to autoimmunity to infection control. For these reasons, along with their accessibility to systemically administered medicines, immune surface proteins and their interactions are particularly attractive therapeutic targets. Although the interaction networks that involve secreted proteins have already been systematically cataloged in the immune system, and more generally across existing protein interaction databases, there remains a substantial underrepresentation of the interactions between cell surface proteins. This is important. So specialized methods have been developed that tackle individual challenges stemming from membrane-embedded surface proteins, such as their typically weak binding affinities, and the low tractability of these proteins for many classical biochemical approaches. These methods, however, generally lack the throughput to systematically characterize whole cell surface proteomes, or have only had success for specific protein families rather than the full diverse spectrum of surface protein topologies and complexes. Thus, how complete our understanding is of extracellular immune receptor interactions has remained unknown. Moreover, many immune receptors of clinical importance have been left as orphans, with their physiological ligands undiscovered, despite, in some cases, decades of study. Without a systematic picture of the physical interactions that link immune cells, any efforts at present to generate truly systems-level views of immune function will remain patchwork at best. So we'll go from here to the first figure. This figure is called Building a Surface Protein Interactome, to enable a systematic survey of the surface protein interactions between immune cells at scales that approach the size of whole cell surface proteomes. We first developed an optimized method for testing binary interactions of all possible pairings of recombinant surface proteins, figure 1a. Okay, so they took biotinylated protein and incubated it either with a plate that had streptavidin attached to it or in a tube with streptavidin tagged with HRP. This then created a prey and bait system. So when the proteins interact together, they stick to the plate and can be detected by HRP. The author's method, the Scalable Arrayed Multivalent Extracellular Interaction Screen, or SVEXIS, simultaneously addresses several key limitations of previous methods to make it possible to screen hundreds of thousands of interactions while consuming minute amounts of protein. And then they look at extended figure 1. So you can see from their extended figure 1 that they have multiple types of proteins, and they identified the optimal binding concentrations. They also looked at their binding curves and the tetrameric prey dilution, so they could see exactly for how long these interactions actually occurred for. So you can see here they did some optimization of their assay, and then as a proof of concept, they said, CD80, for example, binds with CTLA4, so that correlates to this point on this screen, so they can tell what the interaction strength is between all of these proteins, and then they mapped it here. By exploiting multimerization around streptavidin, both immobilized baits and reporter-linked preys for screening can be produced from a single construct instead of two, which is very ideal in being able to produce these high-throughput screens. And the design is attuned for detecting even low-affinity interactions across the range of structural classes that cell surface proteins span, which you saw before in figure 1a, all the different types. We assembled a detailed library that encompassed the full ectodomains of cell surface proteins detectable in previous high-resolution proteomics survey of peripheral immune cells, plus all CD-numbered proteins that were compatible with our recombinant protein platform. So there's a lot of them. The design of each expression construct was tailored to accommodate the structural class of the protein, with 6 different bespoke designs for functionally expressing different receptor topologies and multi-component complexes, shown here in figure 1b. So if we take a look at figure 1b, you can see all of these heterodimer complexes, type 1, GPI anchored, type 2, HLA, multipass, and NKG2-CD94. All of them are able to be expressed with an exogenous signal peptide, covalent biotinylation, recombinant tags, shown here, and this N and C denotes the peptide terminus. Now, this library of 630 different proteins or protein complexes, such as all known integrin combinations, was expressed in human cells, purified, and quality checked. So you've got this in extended data figure 2a and b and supplementary figure 1. We tested all possible protein pairings in both bait-prey orientations for every protein expressed. Extended data figure 2c and supplementary table C. Now in figure 2c, you can see that they tested many things in their screen. Positive interactions identified from this comprehensive primary screen were then retested in a secondary screen with independent protein preparations from a separate cell strain, consisting of an all versus all matrix of 187 proteins. If we look at extended figure 2D, you can see all of these interactions shown here, to give a final matrix of reproducible interactions, shown in figure 1C. So you can see all of these bait proteins here correlating to all of the prey proteins, with a signal strength in the amount of black that you can see, and with the interactions that they did see, they denoted the expected in green and the novel in purple, or in pink, which is a little bit difficult to see, but you can see novel interactions here, 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 here. So that's pretty interesting. Now, in benchmarks Against a hand curation of the published literature in Supplementary Table 3, our screen independently captured a majority of all previously reported interactions at a false positive rate below 1 in 10,000, shown in Figure 1D. So you can see here, as the false positivity rate increases, it doesn't correlate with the true positive rate at a 1-to-1 ratio. There's a much higher true positive rate compared to the false positive rate, and in fact, the area under the curve indicates that it is 0.787. Now, we identified 28 new interactions that were not in our literature curation, expanding the total number of known high-confidence interactions in the human immune system by 20%, shown here in Figure 2E. Now, in Figure 2E, you can see all of these expected interactions are in green, while the novel interactions are in pink. In particular, you've got things like JAM2, self-binding, HLA-E, and VISTA interactions, along with VISTA and HLA-F. You've got LAIR1 self-reacting. You've got CNTN1 with NRCAM and MCAM. So you've got tons of these different interactions that are working together. Notably, these include endogenous non-tumor ligands for the previously orphaned immune checkpoint receptor VISTA, which I just went through, comprising non-classical MHC molecules HLA-A and F. As these measures constitute a highly systematic view of surface interactions for an exemplar cell system, our data also suggest answers to the general properties of surface receptor networks. In the set of immune receptors, 57% of binding pairs are unique, without either protein having another binding partner. Exclusivity is particularly common among proteins that are generally considered to have primary signaling roles, whereas the largest interconnected group features integrins and other adhesion molecules. Next, the authors looked at quantifying total receptor engagement. To validate these discoveries, every interaction was assessed by orthogonal approaches. First, we tested protein binding to the complementary receptor when displayed on human cell surface following transfection with a cDNA that encodes the receptor, shown in Figure 2A. Okay, so you can see here in Figure 2A, on the right here are proteins that are transfected into cells, and on the left are their binding partners. So when looking at the difference between mock interaction with kind of blank protein, like just plain old IgG, compared to, say, JAG1, you can see that there's a distinct curve, and in fact, two different curves. So this indicates that these proteins are binding together. Same with these proteins, because there's a difference in the curves, and these proteins. The only ones that don't look like they have a lot of interaction, I would say, is VISTA and HLA-F, but there is very slight interaction shown here, indicating that these proteins could interact, given that more proteins are present. This was followed by 2 rounds of surface plasmon resonance to characterize direct binding, shown in Figure 2b and supplementary Figure 2. So the surface plasmon resonance shows response units on the y-axis as a measure of seconds, again with these same interactions. For this data, the black is the actual data recorded, the red is the theoretical data, shown by Langmuir model fitting curves. All of the top-ranked 28 interactions were supported by at least one additional method, shown in extended Figure 3C. You can see all of these Sevexis screens. You have primary screen orientation, where the binding signal is high, with the more red it is. So the orthogonal methods are shown here. First off, were they detected by surface plasmon resonance? Secondly, were their kinetics quantifiable? And thirdly, was there evidence of cell surface binding? You can see the validation here is in green if it's clear, light green if it's weak, red if it's none, and gray if it is untested. So that's probably why they said at least 2, probably for this reason here. Now, we realized that we could combine the binding affinities measured in our surface plasmon resonance data with measurements that we methodically extracted from the literature to assemble not only a systematic physical interaction network, but also a uniquely quantifiable one, shown in Figure 2c and Extended Data 4. So Figure 2c is all of their interactions here. You can see that their cell types are color-coded, which is pretty cool. You can look at their binding strength, so you can see this new data set from their data emerging here. And this was verified in supplementary Figure 4, which just has lots of the surface plasmon resonance. Now, we integrated this quantitative receptor interaction network with proteomics expression in leukocytes to gain insight into the patterns of binding kinetics across the immune system, shown in Figure 2D and supplementary Figure 4. Each pairing of two cells, a histogram of inferred interaction data is shown alongside a color shade that indicates the average affinity. They don't really say, but I'm going to assume that the color that they are referencing here is here, with their dark red for high affinity and dark blue for expression, which is why you kind of have some purple overlying here. For example, in their context with antigen-presenting cells, we found that circulating T lymphocytes show a subtle preference for higher affinity receptors when pairing with B cells compared to dendritic cells. Extended Data Figure 5a. So you can see the median interaction shown here, where T to B cell pairing is lower than T cell to dendritic cell, which is an antigen-presenting cell. We also established that the overall distribution of surface interactions has affinities centered in low micromolar range, along with a long tail of higher affinity interactions, shown in Extended Data Figure 5b. So you can see what they are talking about here. In a test of previous theoretical predictions, we found that higher expression levels do negatively correlate with binding strength, although only weakly. So Extended Data 5c and d. So when looking at binding strength, the lower your KD, the higher your strength. So then this curve up here, for example, makes sense in that the lower your number of ligands are, the lower your KD is, which means it has a higher binding strength. So this graph is confusing, at least to me on first pass, because I'd expect that graph to go down. But you want the lower a KD is, the higher the binding strength. So that's why this curve, when it's going up like this, shows that less receptors have higher binding strength. And you can see that that is consistent across all of the data sets shown here. In some of these papers, there's millions of different graphs and multiple ways you have to read them. Sometimes you have to go and look them up specifically to get the details that they need. Of note, we also found that immune activation is accompanied by a broad transition in cellular interaction strengths. Okay, higher affinity interactions predominate in an inflamed state, and these are replaced with more transient interactions in a resting state, possibly to support the need for more dynamic responses when sensing potential threats. In Figure 2, the higher the KD, the lower the binding strength. The lower the KD, the higher the binding strength. So the averages for the downregulated cells, KD values are higher, indicating a lower strength. The activated state for these cells have higher binding affinities, shown here by these lower values. We confirmed this affinity switch in human leukocyte receptor preferences using an independent transcriptomic dataset, shown in Extended Figure 6. So you can see in this extended data figure, most of these upregulated genes after activation. Not so for Tregs, but everything else pretty much follows this higher binding strength and thus a lower KD. So you can see across different data sets with different manipulations and also looking at mRNA frequencies. Our quantitative wiring diagram, if truly systematic to the point of approaching completeness, should make it possible to derive a reductionist model that explains how circulating immune cells associate with each other solely from receptor binding mechanisms and physics-based formulas. We built a coarse yet principled mathematical model that integrates quantitative proteomic expression, binding kinetics, and published cell parameters to summarize the contributions of individual protein interactions to a given cell interaction, shown in Figure 2F. So here is their guide on how they looked at how cells interact. So your surface protein expression, darker blue it is, the higher it is expressed, and the interactions, the darker red it is, the higher affinity. Now, using equations based on the law of mass action, the model then computes how the overall probability of binding between two cell types emerges from distinct spectrum of cell surface receptors that connect them, shown in extended data figure 7. And this is figure 7 that goes through the percentage total binding score, for example, here on the cell-cell pairs, the model of coefficient correlation, and the model of coefficient correlation. So you can see model time step, interactions of these cells, and the predicted model interaction score with the observed interaction score, which has a positive correlation. Although greatly simplistic, our model could still infer the relative frequencies at which human immune cells physically interact with sufficient precision to be consistent with published empirical measurements, shown in figure 2G and extended figure 7D. So we just looked at extended figure 7D, but in figure 2G, you can see this positive correlation between B cells, monocytes, dendritic cells, and T cells. So this positive correlation indicates that their predictions are accurate. The authors next looked at integrating multicellular networks. Now, although our library used circulating immune cells as a source, the immune system traverses a broad repertoire of organs, each of which may be key to understanding the biological role of interaction. We therefore sought to contextualize the interactions of our network by creating an interactive atlas that charts where these receptors and ligand pairs have been detected across single-cell expression databases of human tissues, shown in figure 3A. Here is where you can see figure 3A. Honestly, it's a little daunting. You can see that the cell types are shown here with the interaction lines, and there is a plethora of cell interactions across bone marrow, blood, thymus, lymph node, colon, lung, kidney, and spleen. And our integrated atlas is available here and allows multiple kinds of analysis, which range from summarizing the overall cellular connectivity of different tissue immune populations to inferring which cell-to-cell pairs are capable of carrying out particular receptor interactions, shown in figure 3B and extended figure 8. This is how their 8 single-cell resolution human tissue atlases look. You've got global receptor connectivity, interactions used by each cell type, localization of novel and other interactions, and physical connection maps. And so this is how this looks online. Through this systematic multi-organ atlas, we could determine whether immune receptor interactions proceed through shared structures or are distinct between tissues. We found a recurring motif in which myeloid lineage cells acts as hubs across several cellular interaction networks. Quantified across multiple primary and secondary lymphoid tissues, myeloid cells have consistently higher network centrality scores, shown in figure 3C. So in figure 3C, you can see that myeloid cells in orange have, on average, a higher eigencentrality score across organs, despite expressing similar numbers of surface ligands to other cell types. And if we look at this extended figure B, the myeloid cells on the left, on average, seem to have a higher expressed interacting surface proteins. This suggests that resident myeloid cells may adapt their receptor repertoire to serve as central integrators of local interactions in their tissue niche. Considering the breadth of pathological conditions that exhibit immune dysregulation, we reason that these same integrated approaches could also inform how the physical interactions that we cataloged between immune cells may change in disease. We incorporated paired diseased and reference samples where available in our atlas, which can be used to generate hypotheses on which interactions, cellular or molecular, may differentially appear in diseased states. For example, we could see phagocyte populations shifting a large fraction of their total cellular contacts within the tumor microenvironment of kidney samples, shown in extended figure 8C. You can see a general decrease in tumors and the interaction cell count, including upregulation of APLP2 and APP ligands that we characterized. Our novel receptor interactions could also be integrated with known signaling pathways to infer cellular communication pathways that appeared differentially active in diseased states, which implicated the newly discovered JAM-L interaction in potentially regulating anti-tumor immunity, shown in extended data figure 8D. So if we look at 8D, this doesn't really look like what they were talking about. I think they made a typo. It shouldn't be 8D, it should be 8E, because this is the only thing that even shows JAM-L. So the average expression is shown here, unless this correlates with this shown here. To investigate whether the interactions we discovered in our network manifest themselves under physiological settings in the human body, we finally examined the spatial co-localization of receptors and ligands in a lymph node spatial transcriptomics dataset. Although this reflects only a snapshot of these dynamic cellular populations, both our set of new interactions and the previously reported interactions were significantly more likely to be spatially proximal than randomized networks, shown in Figure 3D. Here in Figure 3D, this is the transcriptomics. You've got interacting neighbor percentage of previously known, novel interactions, and random pairs. So the random pairs are low. The novel and previously known interactions are higher, indicating that they are more likely to be true. In follow-up experiments, we could verify that novel interacting pairs we identified, such as JAG1 and VAS-N, are distributed in bordering regions of immune cell-rich compartments of human lymph nodes, shown in Figure 3E. So you can see with this pretty microscopy picture here, this is a FISH assay, so fluorescent in situ hybridization. You can see CD45 being expressed in yellow, JAG1 expressed in green, VAS-N expressed in purple, and DAPI, which is the cell nucleus, expressed in blue. As you zoom in on the CD45 population, you can see JAG1 and VAS-N being expressed. And if you zoom in even closer, you can see that their co-localization also gives rise to an additional color shown in white, which indicates that they are interacting together. This confirms that these interactions have the potential to occur between immune cells in vivo, shown in extended data Figure 9a. When looking at multiple different protein interactions like JAG1, VAS-N, you can see as well there's this white interaction, PLX-A4 and APP, they're both present, and again, you've got white, so they should be interacting. MCAM and CNTN1, you've got white dots indicating interaction. And here, which is most interesting, I think, is VISTA and HLA-E, where you have tons of white interaction showing that the proteins should actually interact together. So next, the authors looked at assigning functions to the binding targets. We finally asked whether the reagents and receptor interactions that we characterized could have potential clinical use in modulating the immune system. Soluble recombinant proteins with activity to bind specific immune cell receptors have shown promise as immunotherapies. As a first step towards potential therapeutic applications and functional classifications on modes of action, we performed high-throughput cellular phenotyping assays on isolated human immune cells that were treated with recombinant forms of the proteins, identified in our molecular wiring map. To reflect the wide array of interactions and leukocyte subtypes included in our investigation, we adapted an approach for measuring leukocyte activation on cellular interaction phenotypes by high-content microscopy, which captures all major cell populations in a single multiplex experiment, shown in Figure 9C and D. Now, I don't know what's going on here, but extended data Figure 9 only goes up to Figure C, not 9D. So these are the polarization of B cells and NK cells with their most potent activators. B cell seems to be LPS, and as expected from the literature, NK cells are IL-15. That shouldn't happen. I don't know what exactly this is. Extended data Figure 9CD, like it just doesn't exist. I guess that's where we're at. Whatever. Anyway, we incubated pools of human leukocytes in the presence of purified proteins from our recombinant surface receptor library, imaging at 4 and 24 hours to measure changes in both cell-to-cell interactions and proportions of activated cells elicited by each protein, shown in Figure 4a and supplementary Figure 3. We're going to look at Figure 4a. So you can see that human leukocytes were incubated with recombinant protein. They underwent pharmacoscopy, and polarization was shown in a yellow ring, and interaction was with these different colors. Because the function of interactions may not be revealed unless in the proper context, we measured resting leukocytes and those stimulated by low levels of lipopolysaccharide. Proteins from our set of novel interactors elicited diverse responses, including generalized T-cell activation by the formerly orphan TNF-RSF21, as well as the adhesion protein CHL1 and CD320, along with CD58 as previously described, facilitating natural killer cell activation, shown in Figure 4b, and extended Figure 10a. 4b, T cells, LPS, NK cells with 320, C58, cell polarization. Does that make sense? So you've got here just a more complete version shown here with the different proteins. These phenotypes were generally consistent with the cell type expression of each protein's newly identified receptor partners, suggesting direct effects as opposed to indirect mechanisms mediated by an intermediate leukocyte within the mixed pools of cells. Extended figure 10b. So figure 10b shows if significant polarization results for each cell type. False is in red, true is in blue, and across all of these different cell types. The perturbation experiments also provided high-dimensional data on changes in cellular connectivity triggered by infusing soluble receptor binding proteins, shown in figure 4c. 4c is here, so you've got this same sort of setup up top, shown with different cell types with microscopy and processed. The landscape of immune interactions across these conditions provide a rare view into the functional roles of previously described and novel immune surface proteins. This includes large-scale shifts away from cellular contacts between T-cell populations from CD58 along with increased monocyte contacts from SERPA, which is consistent with our previous biological understanding. An inhibition of interactions between monocyte-lymphocyte pairs from SLIT-RK4 and several ligands that we implicated in adhesion, such as NRCAM, CD93, and CDH2 converging on facilitating early B-cell interactions, shown in 4D. So the B cells are in this teal, showing that they are interacting at 4 hours across these different cell type interactions. In line with results reported previously for changes in the interactions of leukocytes after drug administration, infusing our recombinant proteins triggered changes that generally fit within a distinct set of modules of action, shown in extended figure 10c. So here are the expected figures of action. Now again, you can see that the resting states are shown here, and the interaction score goes from blue to red, and the log value are identified all in this figure here. Our in silico model of immune connectivity allowed us to rationalize which molecular changes would have been likely to lead to the observed phenotypes. We compared these experimentally measured perturbations to mathematical predictions as to which cell-to-cell pairs would have the greatest perturbations. Although, as before, there were some gaps in the accuracy of our model for particular conditions. Nevertheless, for most stimuli, we observed significantly greater magnitudes of connectivity perturbations in our experimental data for cell pairs that the model predicted would be most perturbed, shown in figure 4e. So you can see measured interaction change on the y-axis, and if cell-to-cell interactions predicted to be perturbed. So if they were false and low, that means it accurately predicted. If they were high and true, again, accurate prediction. An inverse of this would indicate a false reading. So in conclusion, the author's discussion goes as follows. The immune system is distinctive for being a distributed system. It is not fixed to a single localized organ in the body, but rather is made up of numerous specialized cell types that must adaptively organize their intercellular connections to respond to pathogens and other threats wherever they may appear. We provide a systemic and quantitative view of the cell surface proteins that enable immune cells to dynamically wire their interactions. The receptor interactions that we report in our network each merit further individualized study to characterize their full roles in health and disease. Of particular note are our discovery of HLA-E and HLA-F, but probably not HLA-G, as endogenous non-tumor ligands for the immune checkpoint receptor VISTA. The ability of Visorin to act as a receptor for jagged ligands and immunoglobulin family receptors binding proteins of the amyloid precursor protein family. Notably, members of this same family of non-classical MHC class I molecules that includes HLA-E and HLA-F have previously been identified as key ligands for maintaining innate immune quiescence, which our findings extend to raise the possibility that they may similarly act as regulators of adaptive immune quiescence through VISTA. Our functional screening on blood immune cells further points to pathways worth greater consideration. For example, a role of SLIT-RK4 in lymphocyte responses. While we were preparing this study, independent groups have provided supporting evidence for several of the interactions that we characterized here, including PVR as the ligand for formerly orphan KIR2DL5A and CD146 as an adhesive ligand for CNTN1. Because our physical wiring diagram encapsulates the diversity of surface protein architectures found across all major subsets of leukocytes, it can be integrated with publicly available expression data both qualitatively and quantitatively. The physical interaction landscape that we provide in our single-cell expression atlas offers an interactive platform for deriving insights out of our systemic receptor network, which probably means that this dataset could hold further answers that just need to be explored. Now, although our studies have provided useful views of interactions that are differentially regulated in particular cell types, we provide a systematized catalog of all biological contexts in which an interaction is inferred to be possible, including complementary views directed at particular cell subsets or receptor proteins. This provides material for generating hypotheses about multicellular immune circuits. These same integrated datasets can resolve broader questions in the field. For example, we've shown that immune cell activation is accompanied by broad shifts in the affinity profiles of cell surface interactions, with rapid kinetics predominating during the early stages of immune responses, matching previous conceptual models for optimal antigen sampling and propagating signals to other cells, which switch to higher affinity contacts in an inflamed state to match the changing demand of a forceful inflammatory response. This raises parallels with findings of disease-associated variants that modulate the balance of adhesive receptor affinity. We could also show that the previously theorized link between receptor affinity and the abundance in which low affinity receptors are compensated for by their high abundance may indeed exist, but appears not to be sufficient to account for why certain receptors have evolved the binding kinetic rates we observe. Out of this integrative approach, we could construct a proof-of-concept mathematical model that predicts the behavior of collections of leukocytes from first principles. Although considerable study has been devoted to particular specialized cell-to-cell contacts, such as the immunological synapse, the overall connectivity of immune cells and their dynamic approaches and disengagements have been neglected. Our core model opens up considerable scope for dissecting in a principled way the mechanisms by which immune cells directly associate. In addition, the discrepancies between the model compared to experiments offer opportunities for refining our mechanistic understanding of cell-to-cell interactions by comparing how elaborations to this core model further improve prediction accuracy. So hopefully their core code could be improved on. More broadly, the integrated approaches that we pioneered here for disentangling the immune system provide a framework for future systematic investigations. Using our high-throughput biochemical method for interaction screening, Sevexis, and the strategies that we describe here to characterize interactions by combinations of multiplex cellular assays and genomic datasets, a range of other cellular communities in the human body could similarly be quantitatively mapped. To our knowledge, our study is among the first to systematically map and model how the collective actions of individual receptor molecules through physical laws could explain and predict cellular connectivity on a scale as large as the circulating immune system. Our analysis and the methods we developed provide a template for future studies looking at physical cell wiring networks in detail. From these combined approaches, we may finally begin to disentangle cellular circuits in immunity and beyond, bridging from individual protein molecules to multicellular behavior. Now that we've finished going through this paper and reading it, be on the lookout for the summary video coming out soon. If you like the content here, make sure to like and subscribe. Otherwise, I'll see you next time.